Thank you. First, I would like to thank the Southern Tier Lyme Disease support to invite me to share with you what we are doing. Research on tick-borne diseases at Binghamton University. What kind of research we are doing at Binghamton University at School of Pharmacy. So my research area is really in muscular dystrophy, but when I moved from DC to here, I started hearing about Lyme disease. So I said I will apply the same technology that I used to develop biomarker for muscular dystrophy for Lyme disease as well. So I hear a lot about Lyme disease, and this is a map that I just got from the CDC. And I am pointing to Broome County. Just in 1 year, they diagnosed, these are the cases that were reported to CDC. There are many, many numbers of cases that are not reported. So it was 506 patients affected with Lyme disease in 1 year. And the alarming anaplasmosis that is increasing. And we start seeing babesiosis as well. So if you see the map, there are more tick-borne diseases in the northeast and west east of United States, and in south, we don't talk a lot about it. I didn't hear much about Lyme in Maryland and DC until I moved here. So if you talk about tick-borne pathogens, there are so many pathogens in ticks, the black-legged ticks that lives here in upstate New York. And here I'm just showing one pathogen, the Borrelia, the Lyme-causing bacteria. And you can see that the bacteria is spirochete. And if you look at the surface of the bacteria, there are a lot of proteins there that are specific to this pathogen. And this protein, they help the bacteria to be virulent, infect, and also evade the immune system. So can we detect this bacteria directly by some assay? They are like PCR, ELISA assays. But what I do is mass spectrometry. Detect all the pathogens that exist in a single tick. And I show you the technique in a minute. So I have my student, they collected a lot of ticks from the Shenango Park and State Park. And believe it or not, depending on the season, you drag the rag, you get a lot of ticks. And some season, there are low number of ticks. So we collected both male and female, and you can recognize the female. They have a red color, and the male are black and small. And we bring them to the lab. We put them in a tube, and we put them in 70% ethanol. They die immediately, and they are conserved for a long time. You can freeze them. And then what we do is remove the legs, the head, then we crush the whole tick in a mortar to extract all protein from it. And we have what we call a mass spectrometer. What mass spectrometer does, analyze every single molecule in that soup that we have. Of course, we will identify tick proteins, but also the pathogen proteins with high specificity and confidence. Here I am showing data. It's complex for some of you, but what this mass spectrometer does, it can sequence every single protein in the sample with high specificity and confidence of 99.9 to 100%. So this is the outer surface protein I showed you in bacteria, and it belongs only to Borrelia. But we also detected proteins that belong to other pathogens. So we are sure that that tick has this pathogen, and this. The red peptides are the one we detected with the mass spec, and we can read them. So, like I said, you identify protein from tick, but we had a lot of pathogen in ticks. For here, we got like Rickettsia species, Borrelia, and Anaplasma. Scary, last year we had one tick. It was huge, I still remember it. we analyzed it, has more than 5 pathogens. So this tick is dangerous. So it has rickettsia, and like I said, babesia, when it's there, it takes over. Babesia is not a bacteria, it's a parasite. So doxycycline will not work on this pathogen. But all ticks we collected, especially the female. The male, no pathogens whatsoever, nothing. So all female, they had this Rickettsia buchneri. In the beginning, I thought, wow, 90% of all ticks, they have this Rickettsia. Is it pathogenic or not? But I collaborated with Upstate Medical, and they say it's not pathogenic to human. This is an endosymbiont bacteria. It's called Rickettsia buchneri. It's different from Rickettsia parkeri that causes spotted fever. That's in south. We haven't detected that kind of bacteria in upstate New York. So that's proteins that were detected, and they were very, very abundant in tick, only in females. This is Borrelia proteins, and some season we detected in 1 in 2 ticks, sometimes 1 in 4 ticks, depends on the season, it changes. 2 years ago, or 3 years ago, we detected collected about 70 ticks, and we got more than 50%. And last year, it was less. We got maybe 1 in 5 ticks had Borrelia. So like I said, the buchneri, I was confirmed by our collaborator, Dr. Sinu Wang, upstate, using microscopy. And this bacteria was only in the ovary of the ticks and not in the saliva or guts. And it's believed non-pathogenic to human. It lives with the tick. It's beneficial to the tick, and the tick is beneficial to the bacteria. That's why they call it endosymbiont bacteria. However, the Borrelia and other pathogens, they live mostly in the gut, mid-gut of the tick and in the salivary gland. So when the tick latches into the skin, it decimates under the skin, then infect patient, and then it goes and lodges in the joint, what Dr. Holly was saying, and it evades the immune system. It's very clever with that outer surface protein. And I can show you one of the outer surface protein we identified in the bacteria. It's called the variable lipoprotein surface exposed protein, or VIS. That VIS protein can inhibit the antibody that we develop against the bacteria to kill the bacteria. And it evades the immune system in human as well in the white-footed mouse. Actually, the white-footed mouse is the reservoir for this pathogen, more than the deer. And I think Dr. Ralph Garuto, and Mandy can correct me if I'm wrong, he collected some white-footed mouse. And we are planning to analyze to see if we can detect directly the pathogen in the mouse. But this bacteria is quite clever, or maybe it does that to live in the host. It changes the outer surface proteins. So you can see that in tick we detected a lot of outer surface protein A. Within affect human, that outer surface protein A decreases, and there is outer protein C that increases. Still doing research to understand why did we change from host and ticks. But we collected sample through institutional review board that was approved in 2019 at the School of Pharmacy. We recruited patients or subjects that were affected with Lyme disease. And we did advertisement. Then those volunteers, they came to the school. We draw blood and urine, and we stored them. We have 70 samples stored in the freezer for future analysis or research and maybe collaboration with others. So in 2019, we started the enrollment. When we made the announcement, we had a lot of chronic Lyme disease affected subjects. And we wanted acute patients, so we changed the criteria. So we just said like, where they were affected in less than 3 months, and we collected more samples from both male and female, and age-matched healthy controls. And this is the symptoms that they reported, so we used subject-reported outcome, and you can see clearly that the acute patient or acute subject, they have the same symptoms as the chronic, except that the acute, they have reported, a lot of them reported a headache that disappears in the chronic, but you can see that the fatigue is maintained even 5 years later, some patients. And depression and anxiety was in chronic mostly, not in acute. So this data is from the patient, and we have also asked for the clinical diagnosis if they have. Some they sent us the clinical diagnosis, and some they didn't have that. So these are the areas where they came from, and they visited the school, and we drew the blood. So it was only mostly in the Bloom County, Ithaca, and a few from Syracuse. So we took the serum, urine, and we did that mass spectrometer to look at all the proteins that are altered in their expression to understand what's going on with the chronic Lyme disease patient. So this is comparing the control group, age-matched and gender-matched, to Lyme-affected chronic patient. And we found in the urine that they have pancreatic alpha amylase, mucin, and several other proteins that are altered in their level. And we are validating this data to see if it's really specific to Lyme or it's just autoimmune. So we are analyzing serum and urine from rheumatoid arthritis patient. If they don't see this, then it's specific to the Lyme patient. And recently, this is last week or a couple of weeks ago, we used the kit that we bought. This is for research only. It's not FDA cleared. It's Impiton Lyme, AGM, and IgG test, highly specific test that detect antibody against 37 antigen from Borrelia. So when subject gets infected, the early acute phase, they have high level of IgM, then IgM disappear, and it's followed by IgG. So what this company did, they took like about 37 proteins from Borrelia, they cloned them, and they fragment them, and they make short peptide. If you see them here, and they put them on oil, ELISA, and see which one respond, which one are immunogenic. And the best one, they combine them under one, and they had like 37 of them in one well. So when you do the assay, you will detect any antibody that has a response to Borrelia antigen. So when we tested our serum samples, I was surprised to see chronic patients that reported that they were bitten by a tick 5 years ago still have the antibody. So is it active or dormant Borrelia? We don't know. We still have to validate this data. And you can see the control is nothing. But then the acute, of course, they have a lot. Then we had like more than 50% of the chronic, they still have a little bit IgM, although IgM should disappear. But most of them had IgG. And we are going to validate this and hopefully compare the data to Dr. Ahn with her test. And if the same patient show exactly the same antibody response to the antigen, that means those are real. So we can solve the problem of this chronic Lyme disease that doctors and some other health practitioners don't believe yet. They say like, maybe it's in the head. So this is the new data we have, and hopefully we'll validate it and publish it. And our future work is really was easy to detect the pathogen in tick. But we screened, believe me, we screened a lot of urine sample, and we screened blood, and we could never detect directly the bacteria. I mean, if we do that, we can have direct diagnosis, and we can have specific diagnosis. It could be Babesia, it could be Anaplasma. So you have to do a proper treatment, personalized medicine, and not just give doxycycline to everyone. So this is what I have so far to share with you, and those are mostly the students that did the work. So there are some pharmacy students and some PhD and undergrad, and also our collaborator. And I thank you all for, if you have any question, please.
We thank you. We thank you.
Thank you. And also we thank the people and the patients who participated in this study. Really, they traveled from far. And I was surprised how many we got, 70. The thing we have to reopen this IRB, I just seeking funding for that. I submitted 2 grants to Alex Cohen and to DOD, nothing yet. I'll keep doing that, yes.
I remember in past talks that BU was going to open a big research center and we're working on doing that. How is the progress of that? And 2, is there a place on the BU website that we can check into progress or open opportunities like this?
Yes. If you do Tick-Borne Disease Center, Binghamton University, we have a website. Dr. Garuto was the previous director. He retired. And you know Ralph Garuto, he was like an academician, and he had like 30 years of work or more, I think 40 years of work on ticks and tick-borne diseases. And Mandy was his student, and now she's at Bassett. She graduated, she is a doctor, PhD, not physician. I am not physician either, I am PhD with the research. I get a lot of email, oh, I have Lyme disease, can you tell me if you can diagnose it, to treat it? But I refer them to physicians for that. But there is a website, and I can share that with you. And I think Maggie has the link to that website. And we do have a symposium that we do every year. And maybe in the future, I don't know, we can do one big one. So we invite board research and physicians and clinicians. And I mean, to foster collaboration, that's the only way to understand this disease, especially the chronic. I mean, the acute, yes, you give doxycycline, some patients, they feel better, that's it, and they are done. But some patients, they relapse, and it stays chronic. And we don't understand it. If we understand it very well, we can treat it. That's the key.